That was quick. So I don't need to remind you, but I'm going to anyway. We have an exam in here on Friday. Exam material will cover at least through today. It might cover a little bit on Wednesday. I'll let you know at the end of the day today how far I get, if we can stop or not. Okay? Probably it will just be through today. Two things. One is I have scheduled a review session for Wednesday at 5:15 in ALS 4001, and I will videotape that as I've done before. Okay?
What's the room number again?
ALS 4001. That'll be at 5:15 on Wednesday. Now, second, I've gotten a couple of reports that students, after my instructions to stop writing, were actually coming down the stairwell and writing on their way down. Obviously not cool, since that gives people advantage who are doing that. So as a consequence of that, everybody's going to need to bring their ID next time for the exam. So bring your ID for the exam on Friday, and we will have, I'll tell you more about that at that time. But make sure you bring your ID on Friday, and you'll see how that works in later. Okay, am I clear? IDs have to be with you on your taking your exam on Friday. Okay. I'm going to finish up talking about metabolic control, and then I'm going to talk about glycolysis. So depending on how far I get on glycolysis, that will probably be the last of the material for the exam. So last time I got just a very brief start talking about oxidation and reduction. I'll say a little bit more about that today. But suffice it to say that oxidation is an energy releasing process. And it's through oxidation that chemical energy is stored in the form, excuse me, in the form of triphosphates like ATP. Okay? So oxidation is at the root of that. These processes that involve oxidation are generally what we refer to as catabolic in nature. Catabolic meaning breaking down. So when we're breaking down sugar, for example, we're doing a catabolic process. And that breaking down involves an oxidation. That oxidation releases energy, and that energy is captured ultimately in the form of ATP. We'll see more about that actually next term. Okay. The opposite of catabolic processes is an anabolic process. An anabolic process involves input of energy. It involves reduction. Okay. So when we're making glucose, we are doing an anabolic process. When we are breaking down glucose, we are doing a catabolic process. Okay? Now, as I said last time, cellular oxidations don't occur like oxidations out in the rest of the universe. Cellular oxidations involve electron carriers, so that the electrons that are lost in oxidation don't just go flopping onto another molecule, but in fact they get flopped onto a specific molecule, either NAD or FAD for the most part, and we'll talk a little bit about those. Whether it's NAD or FAD depends upon the reaction, and it depends upon, obviously, the enzyme that's handling that reaction. You're not going to have to know any structures for NAD, so don't sweat that. But you should understand what's happening in what's called a redox reaction. I'm sure you've talked about these in chemistry before, but I'll just refresh your memory. A redox reaction involves an oxidation. It also involves, like I said last time, a reduction. For everything that gets oxidized, something else gets reduced. Because if something loses electrons, something else has to be gaining electrons. The gain of electrons is reduction. Here's a simple oxidation that can occur inside of cells. We'll see several examples of this over this term. Actually, more over next term, but we'll also see an example of it this term. This oxidation occurs with conversion of an alcohol to, in this case, a ketone. Okay? It could be an aldehyde or ketone. It doesn't really matter for our purposes. But an oxidation has occurred. The alcohol loses electrons, and it's giving up electrons to NAD. So the alcohol is becoming oxidized. NAD is becoming reduced. Now people wonder about, well, NAD starts out with a plus sign, and NADH doesn't have a plus sign. What's going on with that? And where does the H come from? Well, the H is just simply a proton that has come out of the solution that is attached to NAD. Okay? Electrons in biological oxidations generally move in pairs. They generally move in pairs. So in fact, this alcohol is losing a pair of electrons to NAD. Well, each electron has a charge of -1, of course. And so we've added 2 negative charges to NAD+, which would, if we didn't do anything else, make it NAD-, right? However, we've gained a proton, and a proton has a +1 charge, so the overall charge of NADH is 0. Okay? All right. So that's what's happening in this type of a reaction. As I said, the alcohol is becoming oxidized, NAD is becoming reduced. If we go backwards, NADH is becoming oxidized, and the aldehyde or ketone is becoming reduced. All right. Well, as I said, there are other things besides NAD that can carry electrons. The most common one that we see in cells besides NAD and NADP, which is related, is this molecule here called FAD. FAD also can carry electrons, and when it carries electrons, it becomes FADH2. Okay? Now, again, we have FAD, which has no charge. We add 2 negative charges of electrons to it. We would have an FAD-, except for we've added 2 protons, and FADH2 therefore has a 0 charge. Okay? So that's the accounting for those. And there are reactions, as I said, that are specific for FAD, others that are specific for NAD, and that's sort of rooted in the chemistry of the reactions partly. Here's a common oxidation type reaction that we see with FAD. We'll see examples of this next term when we talk about the citric acid cycle. In this reaction, here we have what would be a biological acid in essence. It has a single bond between 2 carbons. The oxidation of that molecule involves removal of electrons from that, between those 2 carbons, which creates a double bond. And the electrons go on to FAD and make FADH2. And as I said, this is a common process. This process also occurs during fatty acid oxidation. And we'll see that's a very important reaction. Again, we'll talk about that next term. Okay. And blah, blah, blah. We won't talk about that. We'll talk about that next term. All right. Now, a couple lectures ago, I gave you a definition of a term that I described as an activated intermediate. The example I gave you was UDP glucose. And I said that an activated intermediate was a molecule that had a high energy bond. And it used the energy of that bond to donate a part of itself to something else. Well, here's a molecule that is involved in high energy bond formation. It's known as coenzyme A. And coenzyme A, you probably have heard a lot about acetyl-CoA. Acetyl-CoA is a molecule that is an activated intermediate. It has a high energy, and the energy of that bond between the acetyl group and the CoA group is used to donate the acetyl group to other things. And again, we'll see that next term. There, and we see the CoA being linked to a variety of things. Okay. For those interested in nutrition and things like that, when we look at the B vitamins and various other vitamins, we see that they frequently are coenzymes for reactions. Coenzymes. Now, what's a coenzyme as opposed to a substrate? Well, a substrate is a molecule that an enzyme catalyzes a reaction on. A coenzyme is a molecule that an enzyme uses to do something. In other words, the coenzyme, there's no reaction that's catalyzed on it, but the coenzyme provides a function to the enzyme that the enzyme itself doesn't have. Well, what are some examples? Well, of course, we categorize NAD in this coenzyme category because NAD can carry electrons. An enzyme doesn't do that. So if an enzyme is catalyzing, let's say, a redox reaction, it's going to have an electron carrier as a coenzyme, NAD or FAD, or NADH or FADH2, depending upon which direction the reaction is going. Okay? Other functions that we see that coenzymes have, here's a very common one. We'll talk about it actually next week, and that is biotin. Biotin is a molecule that carries carbon dioxide, and it's involved in what are called carboxylation reactions. Okay? Carboxylation reactions, in which we're putting a carboxyl group onto something else. So carboxylation reactions are very important biologically, and they almost, not always, but almost always have biotin as a coenzyme. Now, I'm not giving you this list to just have you memorize the list because I don't think that's particularly useful, but you will see some of these compounds later that we'll talk about and understand that. You should certainly know what a coenzyme is in general. And there's some molecules, blah, blah, just to let you see what they look like. Okay, there are some non-coenzyme vitamins. Vitamins A, C, D, E, and K don't act as coenzymes, although K does to some extent do that. So that's a little misleading. All right, and that's what they look like. And da-da-da. All right, the last thing I want to say about metabolic control is to talk about reaction types. And your book puts this in this place. It's kind of an odd place to put it, but there's really no other place to stick it, so they decided to stick it into this section. So I'll stick it in this section as well, hopefully not stick it to you. When we look at the reactions that enzymes catalyze, we discover that they can catalyze, we can broadly group all of the reactions that all enzymes catalyze into 6 categories. 6 different categories. These are known as the EC categories for Enzyme Commission that sort of decided this. Right? So there are 6 categories from the EC, and I'm going to briefly talk about them. Okay? The first of these is very easy to recognize. They're known as redox reactions. Well, I've been talking about redox reactions, and not surprisingly, they involve oxidation and reduction. A big clue to redox reactions is they'll always have an electron carrier. When you see an electron carrier present, you've always got a redox reaction that we're talking about. So that is a very, very simple one to recognize. A second category of reaction are known as ligation reactions. And ligation reactions involve joining 2 things together. Well, here is a pyruvate becoming joined to carbon dioxide and making a bigger molecule here, oxaloacetate. This is an example of a ligation reaction. So ligations join 2 things together that were not together before. Isomerization reactions are also fairly easy to see. They involve a molecular rearrangement. So we change the structure of a molecule, and that's all we do in the reaction. Then that is an isomerization reaction. This is a reaction we'll talk about next term again in the citric acid cycle, where we see citrate becoming rearranged to form isocitrate. Okay. And the next one's called group transfer, and group transfer looks on the surface to be a little complicated, but it's really not. Group transfer reactions involve movement of a portion of one molecule onto another molecule. This is a group transfer reaction because we're seeing this phosphate right here in red moving from ATP onto glucose to make glucose 6-phosphate. So we've transferred a part of the ATP onto glucose to make glucose 6-phosphate. So that group got transferred, and that's what's involved in a group transfer reaction. Okay. Hydrolytic reactions are, a good example is here. They're actually a little bit more complicated, but hydrolytic reactions will generally involve water. That involves hydrolysis. That's why they have that name. For our purposes, we will consider them only occurring with water. So when we have a water involved, that's a hydrolysis reaction. The water, as we will see, is frequently used to break apart molecules. So that's a specific thing relative to water. The last one is the only category that I think is particularly difficult to understand, and we don't spend much time on it. I'm not going to be very concerned with it. These are known as lyases. Okay? Lyases are enzymes that split something in 2. Okay? Now, there's actually a much more technical definition of lyases than I'm going to give you here. And I don't think the technical definition is really relevant. For our purposes, something that splits something into 2 is a lyase, and that's it. There's actually some electronic rearrangements that occur that are specific to lyases, but again, we're not going to be concerned about that. This is a reaction we're going to talk about maybe later today, actually, if I get that far. So this is a splitting apart of a fructose 1,6-bisphosphate. Okay, and there's, we don't need to worry about that. Okay, so that's pretty much what I want to say relative to metabolic control. Are there any questions about that before I move to glycolysis? Murmur, 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 murmur. It's funny if you ever get sound effects. You know, you have audiences clapping or cheering or something. One of the things you can always get is an audience murmuring. And after you've listened to those sound effects for a while, you think, how do they do it? How do they get the people just murmuring in the first place? Now I think I know. Yes, ma'am?
So in the hydrolytic reaction, is that like the serine protease? We use water to...
So her question is, a hydrolytic reaction, would a serine protease be an example of that? The answer is it absolutely would, yes. Okay, well, let's turn our attention now to metabolism. We're going to be in metabolism for a while. We're going to talk about metabolism for the rest of this term, and we'll be talking about it for the first half of the next term. So we've really built a foundation now so that we can start talking about the reactions involved in keeping cells alive, metabolism. All right, well, metabolism is a subject that I think it's very easy to get lost in all of the reactions. And I'm not going to make you memorize all these structures because I don't think that's overly productive. Any structures that I've asked you to memorize, like glucose or fructose, whenever they appear in a name, as we'll see, those are fair game. If I say glucose 6-phosphate, all you have to do is put a phosphate on number 6 of glucose and you've got it. So if I ask you something like that, then of course you should be able to draw the structure of anything I've already expected you to know. But I'm not going to ask you to draw, for example, the entire pathway of glycolysis. I don't think that's particularly useful for you to be able to understand what's going on. It's more important that you see the bigger pictures with respect to the metabolic pathways and how they are controlled and how they affect cells, because you're going to understand, I hope, a lot more about how your body works by understanding these metabolic pathways. Glycolysis is the first one, gluconeogenesis is the second one, and then we'll talk about glycogen metabolism as the third and fourth pathways. So we've got some things to discuss. By the way, you'll see that at this point in the term I also have separate notes that I have written, okay? And the notes are summaries, et cetera, et cetera. These are just there for your additional information. I'm not trying to give you more material to read or memorize or any of that. It's just there as a reference for you if that helps you to understand the material. Okay. Well, when we think about glycolysis, glycolysis literally means break down glucose. Glyco referring to sugar, the sugar in this case being glucose, lysis meaning tear apart or break down. In glycolysis, glucose is converted from a 6-carbon sugar to ultimately 2 3-carbon molecules, okay, right here. This process of glycolysis is right there, that one step. Well, glycolysis actually has 10 individual steps of its own. And what you can see in this figure is that pyruvate is not an endpoint. In fact, in metabolism, we don't generally see endpoints because pyruvate becomes a substrate for other pathways. So the number one thing I want to impress on you here is that pathways are things that are man-made inventions. In the cell, there's no dividing line that says, okay, this is glycolysis, and this over here is pyruvate metabolism, and this over here is acetyl-CoA metabolism. There's no such rules, okay? So all these things are happening. Now, in the cell, we do see compartmentalization. We do see some reactions will only occur in the mitochondrion, for example. All right? But those are physical limitations. They're not pathways. Pathways are human beings' inventions. Are we clear on that? There's nothing magical about these. We've just decided in our definition that glycolysis ends at pyruvate. Okay? All right. Well, that's where we're going to start this. So glycolysis ends at pyruvate. Pyruvate is an interesting molecule, and I'll say more about it as I get going along because pyruvate really has several possible destinations after it's been synthesized. And the destinations it has gives us our first example about how cells change their metabolism based on conditions. Which direction that pyruvate goes depends upon the conditions it encounters and also upon the cell type in which it's found. All right? This top pathway right here is very popular if you're making beer or wine because bacteria and yeast use this pathway. We can't go that pathway. All right? We go this pathway under the same conditions that bacteria and yeast have. And so therefore, what we see on the top is fermentation going to ethanol. Fermentation in us produces lactate. And yes, we do ferment. We just don't produce the good stuff. Okay? If we're not fermenting, all aerobic cells will produce oxidation of pyruvate, ultimately to make carbon dioxide, and we'll see how that occurs again next term. Okay. Now, let's just take, and I know this is not the best figure in the world, it's straight from your book, and this is one of the reasons I recommend that you print out the figures before you come in, because you're probably not going to copy this down as I'm talking today. What I want to do now is go through the reactions of glycolysis with you and say a little bit about the relevant information in them. You do not have to memorize the structures in here, and I do think that you should be able to say fructose 1,6-bisphosphate goes to glyceraldehyde 3-phosphate and DHAP. So in other words, I think you should be able to give me the names of the products of all the reactions. All right? Those are reasonable. And you'll see that there's a link to them. The product of one reaction is the substrate for the next reaction. That's true of metabolic pathways. So in the very first reaction of glycolysis, glucose goes to glucose 6-phosphate. In the second reaction of glycolysis, glucose 6-phosphate goes to fructose 6-phosphate. So there's a link between them. That is, the glucose 6-phosphate was the link for both of those reactions. Well, so anyway, I'm going to go through and describe these to you. You do not need to know all of the enzyme names, although to be honest with you, they actually, if you know the molecular names, they're all pretty straightforward. Okay? They're pretty straightforward. But I will tell you which enzymes I think you need to know as I go through. Okay? We on target? Everybody's looking glum like, man, it's going to be a lot of stuff now before the exam, right? Okay. Well, we'll try to keep it as general as we can. All right. Well, I want to metabolize glucose. The reason I want to metabolize glucose is ultimately I can get a decent amount of energy out of glucose. And glucose is freely flowing through my veins, arteries, and my cells therefore have a ready supply of glucose. So it's something that I can use as a very quick energy source and an immediate energy source. So glycolysis is very important for that in that respect. We start out with the very first reaction. Glucose goes to glucose 6-phosphate. If you want to abbreviate glucose 6-phosphate, you can abbreviate it as G6P. So glucose goes to G6P. Now, this reaction is one of the reactions for which you need to know the name of the enzyme. The enzyme here is an interesting one. It's called hexokinase. Again, the name of the enzyme tells you what it does. Hexo meaning 6, kinase meaning phosphate. Hexokinase puts a phosphate onto 6 carbon sugars, like glucose. Will it work on other 6 carbon sugars? The answer is yes, it will. It actually has a fairly broad range of sugars on which it will put a phosphate. Notice that ATP energy is required for that addition of phosphate. ATP energy is required. Okay? The phosphate comes from ATP. That's that group transfer reaction I showed you just a few minutes ago. All right? Now, why do I want you to know this enzyme? The reason that you should know this enzyme is because this enzyme is important in controlling glycolysis. There are 3 enzymes, it turns out, that are important for controlling glycolysis, and this is one of them. Now, this reaction is an interesting one in the sense that we can try and run this reaction without ATP. That is, what if we just put phosphate into the reaction instead of ATP? What would happen? Well, it turns out that we can get that reaction to go slightly if we started with equal amounts of material. Okay? Slightly in the sense that we don't destroy all of the glucose 6-phosphate if we started with that. It tells us that energetically, if we just put phosphate into there, that this reaction is not energetically favorable. I can't make hardly any glucose 6-phosphate if I just simply use phosphate instead of ATP. Okay? On the other hand, if I run the reaction with ATP, I discover that this guy has a delta G 0' that's fairly negative. Okay? Well, a fairly negative delta G 0' means that if I start with equal concentrations of products and reactants, the reaction is going to go forwards. Okay? Meaning it's a favorable reaction. So what I've done with ATP is I've converted a fairly unfavorable reaction to a fairly favorable one. And you go, well, duh, there's energy in ATP. And the answer is, yeah, I realize that's a duh, it is. But it tells us that this process that I've just described to you is a very important one metabolically. It involves what we call coupling. Coupling involves the hydrolysis, the use of a high energy intermediate in a reaction to drive an energetically unfavorable reaction. So I've been telling you then that the addition of phosphate to glucose is energetically unfavorable if I just do it by itself. Yes, ma'am?
Can you repeat the coupling?
So coupling involves use of a high energy molecule, okay, let me back up and give you a better definition. So coupling involves joining an energetically favorable reaction with an energetically unfavorable reaction to make the overall sum of the reactions favorable. What are the two reactions? One of the reactions is the hydrolysis of ATP. That's energetically favorable. The energetically unfavorable reaction is the addition of phosphate to glucose. So when I put these two together, the entire thing becomes energetically favorable. That's coupling. Okay. I'm not going to ask you to define coupling if that's what you're worried about. But the concept of coupling is important. I use a high energy ATP to put that phosphate onto glucose. Okay. What does that tell me about the energy of glucose 6-phosphate compared to glucose? It has higher energy. It has more energy than glucose does. That's why I'm having to put energy into it. Okay? So I need that energy from ATP in order to drive this reaction and make it happen. If I don't have that energy from ATP, the reaction will not occur to any significant extent at all. Okay, well, that's the first reaction. Hopefully they don't all take that long, and they don't. The second reaction of glycolysis is fairly straightforward. It's catalyzed by the enzyme phosphoglucose isomerase, and no, I don't expect you'll need to know the name of that, which means nobody will learn it. In this reaction, it's a simple isomerization reaction in which we are converting glucose 6-phosphate into fructose 6-phosphate. It's simply a rearrangement of the molecule. This reaction is, and by the way, do you need to know the energies of the reactions? The only reactions that you need to know the energies of, I won't ask you for a number, but you should know which reactions are energetically very favorable and energetically very unfavorable. And I'll tell you those as we go through. This one is pretty much in the middle. It's not one that is energetically favorable or unfavorable. And when we see a reaction like this, it will have a delta G0 prime fairly close to 0. And because it's fairly close to 0, it means it's readily moved forwards or backwards. Yes, ma'am?
So since the first one is coupled to an energetically favorable reaction, does that one count as energetic?
The first one counts as an energetically favorable reaction. That's correct. Good question. Okay. All right, so now we've got fructose 6-phosphate. We're going to need to put another phosphate on there to make this reaction go forwards. The addition of another phosphate requires another ATP for the same reason we needed one to start with the glucose. That is, if we're going to make fructose 1,6-bisphosphate, that's a fairly high energy molecule. I have to use an energetic molecule to add that phosphate, and voila, ATP is available. ATP provides the energy necessary for the synthesis of fructose 6-phosphate. The enzyme that catalyzes this reaction is a very interesting enzyme. And yes, it's one that you're going to need to know. It's the second one that regulates something in glycolysis. Now, most metabolic pathways have like one enzyme that control them. Glycolysis has 3. And it turns out that every single one of them is involved in an energetically favorable reaction going forwards. An energetically favorable reaction going forwards. Well, if a reaction is energetically favorable going forwards, what does that mean about the backwards reaction? It means it's energetically unfavorable. Okay? All right. This enzyme, phosphofructokinase, which you are more than welcome to abbreviate PFK. And by the way, you can call fructose 6-phosphate F6P if you want. And you can call fructose 1,6-bisphosphate F16BP. That's fine. Okay. Phosphofructokinase, or PFK, is the most important regulatory enzyme in glycolysis. We're going to see, and we're not going to talk about it until Wednesday, but we're going to see that PFK has a lot of different ways of being controlled. That means, therefore, that there's a lot of factors that influence whether it's active or not, and a lot of factors, therefore, that influence whether or not glycolysis is running. Well, why is this the most important one? Well, if I stop the reaction at PFK, what's going to happen to the entire pathway? It's going to stop, right? Okay? Because if I've got everything blocked, I want to say plugged or blocked. Maybe if I said plugged. If I've got everything blocked right here, all right, then there's nothing down here to go forwards. So that blocking of one enzyme still effectively shuts off the entire pathway. As I said, this reaction is energetically favorable because ATP is present. The product of this is the molecule fructose 1,6-bisphosphate. You can see the two phosphates on there, and it's the substrate for the next reaction that is also a very interesting reaction. Okay. Now, I know there's a sort of a parallel to the signaling pathway. This goes to this, goes to this, goes to this. So that's just a sequence that you're going to have to get used to. You're going to have to understand. The next reaction is the place where the 6-carbon sugar, in this case fructose 1,6-bisphosphate, gets split into 2 3-carbon units. So this is a lyase reaction I'm getting ready to describe to you. Splitting it in half. And this reaction is interesting in several respects. First of all, the name of the enzyme that catalyzes this is the only enzyme name that doesn't really tell us an awful lot about the molecules involved. The name of the enzyme is called aldolase, A-L-D-O-L-A-S-E. Aldolase catalyzes the formation of dihydroxyacetone phosphate, also known as DHAP, and glyceraldehyde 3-phosphate. And here's where I differ from your book. Your book uses the abbreviation GAP. I don't like that abbreviation, so I call it G3P. So please call it G3P if you're referring to glyceraldehyde 3-phosphate. Okay. Now, dihydroxyacetone phosphate, DHAP, and glyceraldehyde 3-phosphate, G3P. They're shown right here. Okay. Now, the aldolase reaction is interesting because this reaction I have just described to you is energetically very unfavorable. Has a very positive delta G0 prime. Now that seems a little odd, doesn't it? You've got a metabolic pathway. You've got right in the middle of the metabolic pathway, you've got a reaction that is energetically unfavorable. Don't you think cells are going to need to run glycolysis? The correct answer is yes. Nobody has a correct. Everybody's staring at this today, okay? Do we need to stretch? Okay, let's stretch. Stretch, stretch. Everybody get up. Come on. Stand. Okay. This reaction, the aldolase reaction, is energetically very unfavorable. Okay? Yet glycolysis occurs inside of cells. Now notice I said it was the delta G0 prime that was very positive. Does the delta G0 prime determine the direction of a reaction? No. It influences it, right? So if I have a very positive delta G0 prime, what do I have to do to make the overall delta G be negative? It means the log term has got to be more negative than that positive term, right? What makes the log term more negative? I heard several things, I'm not quite sure, so I'll say. All right, so what makes the log term more negative is the fact that we've got more reactants than we do products. And that's a very, very important consideration. We're going to see later that cells actually manipulate that in a very interesting way. But suffice it to say, if we have more reactants than products, we can force that reaction forwards. And that's exactly what cells have to do. Okay? That's exactly what cells have to do. They have to make this reaction go forwards. If I'm sitting around and nothing else is happening, this reaction isn't going very far. What starts happening is reactants start building up. And as they're building up, they're getting closer and closer to making this reaction becoming more favorable. At any given time, your cells already have that sort of ratio built into them. They're already close to that 0 point because they've already built up an awful lot of reactants. Tipping that balance makes things go forwards, and we'll see how that occurs later. Okay. The next step is what you see, what's shown here is a sideline. And the sideline is not drawn very well. As I said, the aldolase produces both of these two molecules, but then once both of these two molecules are produced, one of them, the DHAP, gets converted back to glyceraldehyde 3-phosphate. This involves another enzyme known as triose phosphate isomerase, TPI. TPI converts DHAP into G3P. It's just a simple isomerization is all that's going on. Okay? Well, the beauty of that now, it becomes very easy to track everything because at this point we now have two glyceraldehyde 3-phosphates. And every other reaction we have two identical molecules. Yes, ma'am? So the enzyme that converts DHAP into glyceraldehyde 3-phosphate is known as triose phosphate isomerase, or TPI. Anybody remember where you've heard that enzyme before? Good pop quiz. What's that? You've heard it once before in the term. Anybody? 1 point of extra credit to the first person that tells me. In what respect? What was I talking about? It wasn't when. I want to know what. Or last Monday, right? Nice try. Okay, I will tell you. It's an example of one of the perfect enzymes. Oh, yeah, the perfect enzymes. I knew he was going to do this, right? It's a perfect enzyme. It has a very large ratio of kcat to km. And the reason why is because the intermediate that this reaction uses is very unstable, and if it sits around for any period of time, it will produce an undesirable product. So the enzyme appears to have evolved perfection as a way of reducing the time that unstable intermediate is present. Very cool phenomenon that happens. Okay, glyceraldehyde 3-phosphate is the product of this reaction. At this point, we have two glyceraldehyde 3-phosphates. So as I said, we have two for everything going on now. You'll notice we've gone through all these reactions. We haven't produced any ATP. In fact, we've used two ATPs. We haven't had any oxidations going on. So where's all the energy in glycolysis? Well, the energy in glycolysis is in the last half of the reaction, or the last half of the pathway. The first of these is an oxidation that occurs on glyceraldehyde 3-phosphate. This oxidation is catalyzed by the enzyme glyceraldehyde 3-phosphate dehydrogenase. Whenever you see the term dehydrogenase in an enzyme name, we're talking about an oxidation reaction, a redox reaction, same thing. Okay? This reaction I'm actually going to show you because you have to see the oxidation that's occurring. Okay? So, when we look at that reaction, here's what's going on. Glyceraldehyde 3-phosphate, they call it GAP, I call it G3P, okay, plus NAD plus phosphate. Notice we have phosphate going on here. We didn't put ATP in here. Makes 1,3-bisphosphoglycerate, or 1,3-BPG, plus NADH. Now, we started with something that had 1 phosphate. We ended up with something that had 2 phosphates. Why didn't we have to use ATP? He says favorable reaction. Why was it a favorable reaction?
Wild guess.
Wild guess. Oh, that was a wild guess. There's not a wild guess that goes with that. Okay.
No.
Okay. It is a favorable reaction, but why is it a favorable reaction? What makes this addition of phosphate favorable here, but it wasn't favorable when I had glucose or fructose 6-phosphate? Why is this one favorable? We've been talking about it. What is it? Oxidation. Thank you. Okay. This guy is getting oxidized. Okay. You'll see that this is an aldehyde. This is an ester, meaning that there was an acid that was formed. Oxidation of an aldehyde to an acid is an oxidation, and we see the reaction going on right here. Okay? Here's the aldehyde. First step, we get the acid. That's the oxidation. Second step, we get the addition of the phosphate. The energy of the oxidation is what drives the attachment of the phosphate. Without that oxidation, we have an energetically unfavorable reaction. Yeah.
So oxidation, you're losing electrons.
Yep.
You're going from an H to a PO3. Are you getting a lot more electrons? You're adding electrons to the molecule.
Well, you're adding an entirely other molecule, but the carbon itself has lost electrons. That's the important thing. We're not counting the total number of electrons in the molecule, but the number of electrons relative to that carbon. Okay? Now, so in going from here to here, there's an oxidation that has occurred. So these electrons have, in fact, been lost. Okay? That's where we see they're picked up here by NADH. So the oxidation reaction drives the addition of the phosphates. So we don't need ATP in this reaction because we have an oxidation making this whole thing possible. All right. Well, this oxidation now has produced a high-energy intermediate. This high-energy intermediate is known as 1,3-BPG. Does that sound like a molecule we've talked about? Okay. It's not the same molecule, though, is it? All right? And it turns out it's not even directly connected to it. We'll see how it comes into play in just a little bit. All right. The next reaction is the first time we make ATP. We're actually making ATP in this reaction. And this reaction, this high-energy phosphate on the glycerol on the 1,3-BPG is transferred to ADP to make ATP. Okay? Now, somebody asked me a similar question about this recently. Well, why do we call this a kinase? Okay? We call it a kinase because, again, a kinase puts phosphates onto things, and the phosphate is moving from here onto here. That is a kinase. Okay? So we're putting a phosphate directly onto ADP to make ATP. Anytime we directly make ATP in a reaction like this, that is transferring a phosphate from a high-energy molecule onto ADP, we're doing what's known as substrate-level phosphorylation. Substrate-level phosphorylation. Now, why is that important? It turns out that substrate-level phosphorylation is one of 3 ways that ATP can be made. It turns out to be the least important of all of them. This is the least important that in terms of abundance of all 3 ways that ATP can be made. The other ways that ATP can be made include two other different types of phosphorylation. One is called oxidative phosphorylation, which our mitochondria do very abundantly. And the other is known as photophosphorylation, which plants do in photosynthesis. This one is the least abundant of all three. Nonetheless, it's important because it's giving us some energy, but it's not the most important source of ATP. Now, I'm going to finish with the lecture with this next thing that I think will teach you something about your body. Okay? And it's this reaction right here. I'm not going to talk about these until Friday, and you won't be responsible until Wednesday, and you won't be responsible for the last two reactions on the exam. You're only going to be responsible through this reaction right here. Now, this reaction is very interesting. Oh, by the way, that last enzyme where we made the ATP, actually, never mind, forget that. Forget the enzyme. You don't need to know it. All right. This enzyme is interesting. I'm not going to hold you responsible for knowing it. It's called phosphoglycerate mutase. And whenever you see the word mutase appear in an enzyme name, something interesting is going on with it. Mutase enzymes have a very interesting mechanism that they use to make their molecules. All right? Well, it looks like this enzyme is simply taking this phosphate from position 3 and moving it up to position 2. We're going from 3-phosphoglycerate to 2-phosphoglycerate. And that's the net result of what happens. But the mechanism is not what I just described to you. The mechanism by which this occurs is mutases will make an intermediate that has a phosphate on both position 2 and position 3. So the first thing it does, it puts a phosphate on position 2, and then it removes the phosphate from position 3. So it's a two-step. Put a phosphate on here, take the phosphate off the other one. So the phosphate that's on position 2 is not the one that was down here. Now, that intermediate was very interesting, because that intermediate turned out to be 2,3-BPG, that very important molecule for hemoglobin. Now, let's think about this. When cells are actively respiring, they are going through glycolysis very rapidly. It turns out that that intermediate, 2,3-BPG, is stable and can be released by the enzyme. The enzyme doesn't hang on to it tightly. Thank goodness it doesn't. Because when it releases 2,3-BPG, what's the signal? I'm going through a lot of glycolysis. I'm metabolizing a lot. I need oxygen. Very cool. Okay? Thanks to the stability of that intermediate, your hemoglobin can read if you're going through a lot of glycolysis. The more glycolysis you're going through, the more 2,3-BPG will be released, and more oxygen will be delivered to that cell. I think that's really cool. And it happens because of the mechanism of a mutase. Okay, now, the reason I said in class that 2,3-BPG can be broken down very readily is it can, because an enzyme that is a phosphoglycerate mutase that is just sitting around can pick up that 2,3-BPG later as the hemoglobin is dragging it back to the lungs. It grabs that 2,3-BPG and it breaks it down. That's why the 2,3-BPG doesn't generally make it back to the lungs intact unless you're a smoker, in which case there are other reasons why that happens, and I won't talk about those today. Okay, so exam material covers through today. Don't forget, on Friday you will be bringing your IDs. I will remind you of that on Wednesday. Review session on 5:15 on Wednesday. And question? I haven't gotten to it yet.
Okay, so we
Yep.
don't need to know.
Yep. Hey Andy, how you doing?
It's going to be on the file?
It will be on the file, you betcha. Are you going to take my file?
No. I already passed the course.
There you go. I never have to do it again. Microphone's working great.
It wasn't this morning.
Really? You know, are you turning the mute off on that second thing?
No, someone before
You might
me
try
does.
that. You might try that because, oh, let me close this. Because I'm finding the last 3 or 4 times it's worked perfectly. I haven't had any problems at all.
I
So,
had major problems this morning because some idiot before me pressed some buttons that I had to get them to remotely undo.
yeah. Wow. Hang in there. Hi.
Quick question.
Yeah.
On that mutase,
Yeah.
is the reason that it's not an isomerase because it does
Right, isomerases rearrange.
Okay.
Mutases do that
kind of
two-step.
mechanism of
Yeah, yeah. It's mechanistic as to why it does that. Isn't that interesting, though?
And do you have a brother that's an astrophysicist?
I don't. Nope.
There's a Professor Ahern from Maryland University on like intro.
Oh, okay. Yep. Nope. I don't. I have a brother, but he's not an astrophysicist. So, take care. Yeah.
I talked to you earlier in the term about taking the midterm early.
Oh, that's right. I saw you. Okay.
Yeah,
Sure.
I